Hi, Mike. Hi, Ken. Nice to meet you.
Nice to meet you.
Yeah, well, Ken, thanks so much for joining us. This is really a huge pleasure for me. As you know, your work is incredibly important for the field, and I want people to be able to hear you talk about it. I was reading your papers for years. Being able to work with you on that 2002 paper in the chicken frog was a huge pleasure for me.
I love that paper.
Yeah. Have you heard anything from Thorleif? Like, how's he doing?
As far as I know, he sort of left research and has gone full bore clinical.
I see.
He was trained he had an MD PhD sort of thing. So I think he's just doing clinical research. And so I have not been in touch with him, no. Yeah.
Aastha, we cannot hear you. Are you muted? Try.
Oh, can you hear me now?
There we go. That's it.
Oh, perfect. Okay. Okay, fantastic. If we're ready, we can get started. Ken, thank you again for your time. Well, we're digging into the origins of bioelectricity and trying to understand the history of the field from your perspective as well. But maybe we can get started with you sharing a bit more about your background.
Okay. My training was in physics. And so I was an undergraduate in physics and started graduate school in a PhD program in physics. And at the end of the first year, I passed my qualifiers and was ready to start my research in physics. And I spent a summer in a laboratory that I was going to work on. It was a nuclear laboratory doing low energy nuclear physics. And that summer, I had a chance to talk to the postdocs and graduate students, and these were incredibly bright people, hardworking. They could do the theoretical analysis. They could build their own electronics. They could do high vacuum plumbing, blah, blah, blah. They were really skilled people. But everyone that I talked to felt that their actual research project wasn't very interesting, that it was not exciting. It was adding on a little bit to an already well-established story. And that, I didn't like that. I thought of myself burying myself down in the bowels of the earth with an accelerator for 5 years to do something that nobody cared about. That didn't do very well for me. I didn't like that. So I had read just enough biology. It was funny. I was in the Peace Corps in the Philippines, and we were given a locker full of books that we were supposed to just leave behind. And one of those books was a little paperback called The Genetic Code by Isaac Asimov. And I knew just enough to be able to read it and make sense of it because it was written at a simple level. And so I was aware of these monumental changes that were going on in biology. This was in 1963 or 1964. Okay, so the genetic code itself had just been worked out. So anyway, when I got discouraged with physics, I decided to see if there was some place in biology that would be useful for me in the Purdue biology department. And I was lucky because that department had a strong history of people coming in from physics. Michael Rossman was there. Was an X-ray crystallographer, a very distinguished one, and he had come into biology from physics. And a number of other people were there that had come in, had joined it, biology from physics. And I went to one of them, a guy named Bill Pack, who is a Drosophila geneticist of considerable note. He just set about mapping every mutant that he could in Drosophila by looking at their retinal function. He put little electrodes on the eye of the fly and looked for deviations in the kind of response, the electrical response that they got, and catalog mutants that way. And many of those turned out to be extremely interesting and important 20 years later. At any rate, he couldn't take me in his lab. So he told me, he said, well, there's a new guy, a new faculty member here that's, I think, looking for graduate students. And that was Lionel Jaffe. And he sent me to Jaffe, and Lionel was willing to overlook the fact that I hadn't had a biology course since I was in high school, and invited me to join his lab. And I did. And it was so different than what I was going to do in physics. And I was immediately able to be somewhat productive. And I set about trying to learn some biology. And with some, I actually I call myself a biologist now, although I still haven't ever had a course in biology. And that's how I got started and of course, Lionel was interested in aspects of bioelectricity, and I was able to contribute there, and that's what got everything started.
Wow, that's amazing that you didn't have any biology background and got into it with physics. But it is interesting to me that it seems like quite a few of the scientists in the bioelectricity field came from different backgrounds. I know, Mike, you also had computer science, and it's just such a multidisciplinary field. I'm curious, when you first entered the quote unquote field of bioelectricity back then with Lionel Jaffe, did you know anything about it, especially given that it was the era of the genetic code? What was the state of bioelectricity back then?
I think it was considered to be a curiosity. And there certainly was at that point a feeling that, well, Lionel had a phrase for it. He called it a coliform vision. Coliform referring to E. coli. And he said that, and I was an innocent bystander. I didn't know these disagreements and everything, but I learned. He felt that the people who were studying developmental biology at that time had been convinced that if you simply learned everything about E. coli and metabolic pathways in E. coli and everything, then you would understand development and everything about development even up in the humans. And he vigorously and colorfully disagreed with that view. And I began to learn then a little bit about what I guess I can call the politics of the science from that point of view. So that's how I started finding out about what these differences were.
And how big was, I guess, Lionel's team or people who were also thinking in terms of how Lionel Jaffe and you and some of the others were thinking about it?
How big was the lab? Is that what you're asking?
Or not just the lab, but in terms of like how big was the group of bioelectricity back then in general?
Well, broadly speaking, we were trying to put bioelectricity on a really firm basis. And so we looked to the neurophysiologists as for our inspiration and for how to proceed. So, for example, people like Hodgkin and Huxley and Katz, the people that had won Nobel Prizes for understanding how a nervous signals are generated, that we considered them to be that we were a branch of them, and we were trying to be as rigorous as they were, and we were using many of the same techniques that they developed. So there was a fair number of people. There were not many people, however, in developmental biology who were in fact doing that the way we were. It was a relatively small group.
Can you talk about some of the research you initially did when you first entered the lab?
Sure. My first project was just to, well, the laboratory, Lionel's laboratory at that time was focused on how polarity emerges in an algal egg from the brown algae, the fucus and what was then called Pelvicia, now called Sylvicia. And this was an interesting organism, and it's the first thing I knew and worked with, in that when the egg is released from the plant, the alga, and then it gets fertilized, and their reproductive cycle is very animal-like. You know, you release eggs and sperm, and the sperm binds to it and fertilizes it. The first thing that this egg does is settles down, and it chooses its basic axis. There is no preformed axis in these eggs, so it's all developed, and this takes hours. And the usual way that axis is formed is in response to unilateral light. So if you can imagine the egg sitting in a tide pool with the sun above, well, it develops its rhizoid, which is its root-like structure, on the opposite side. And that's directed by the light. So these cells are extremely sensitive to unilateral light, particularly blue light. And we were interested in what were the mechanisms by which the cell perceived the light and then responded to it. This all occurs, and this axis is formed, and it becomes asymmetrical, and starts growing out at one end while it's still a single cell. It hasn't even yet divided. And when it does divide, the division is perpendicular to that axis. So the result is you get two highly unequal cells formed that have different developmental fates. And this all happens in a way that you can control it. The experimenter can, just by using light. And Lionel was interested in ionic changes and membrane potential changes. So one thing that I did is I started measuring ions moving, potassium, sodium, calcium, chloride, moving into and out of the cell. And I got experience using, radioisotopes and doing these kinds of measurements on these cells. But the first significant piece of work that I did, I think important piece of work, was I figured out a way to take a very thin piece of metal, which had someone else made. They were made as filters for rocket engines. And they had perfectly round etched holes in this very thin piece of metal. And I found that I could fill every one of those holes with an egg. And then they would seal themselves into it. So the result was you create a sort of an artificial epithelium. It was a way I was putting the eggs in parallel with each other. I didn't have the techniques for measuring ion movements into and out of the egg in a single cell. So I had to put 25,000 of them in one of these sheets. And then the experiment was that I would take two such preparations like that were in a chamber where I could flush fluids through them. And on the underside of each one, the underside because it was more uniformly exposed and there would sometimes be extra eggs on the top, I'd take two such preparations and add radioactive calcium to the underneath side of each one. But one would have light coming from one direction and the other one would have light coming from the other direction. So I was forcing them to polarize one way or the other. And startlingly enough, the side that was on the shaded side, the dark side, which is the one that's going to grow into the rhizoid end, there was a huge difference in the calcium, radioactive calcium influx into that side versus the other side. It's about a 5-fold difference. And so we had direct evidence that, and this is before any morphological changes have occurred. We had direct evidence then that calcium channels were being activated on the dark side. And that was absolutely necessary for forming the axis. And that paper, my photograph of the screen, of a filled screen, was on the cover of Science. And it was a nice little article in Science. And I think that paper went a long way for establishing my career.
And do you think, so in that assay, do you think that, and maybe this is also more for this other experiment that you guys did in a capillary, but do you think those eggs were mostly communicating with the environment, or do you think they also communicate with each other in that?
I think they were acting independently of each other. I think they were all just responding. We know I subsequently learned a fair amount more about this process. Interestingly, it turns out, well, Lionel had done some just absolutely gorgeous work showing that the dichroic axis of the photoreceptor was parallel to the plane of the membrane, of the plasma membrane. And I then got evidence that the protein that was involved was in fact an opsin-like protein. And we did measurements. I worked with some people in Italy who use mass spectrometry and so forth, and they could really measure these what they were looking for, the opsin. They were looking for the chromophore, which was the same as the chromophore in the human eye, retinal, and they found that it was absolutely packed with retinal. And we estimated that the membrane is practically a crystalline array of opsin-like proteins. And so the action starts on the illuminated side because these cells are optically very dense and they absorb 98% of the blue light that's impingent upon them. So one end gets highly, receives a lot of light and a lot of excitation, and that's the bright end, the illuminated end, and the dark end then somehow or other becomes the growth point. But we found, first of all, the calcium is involved there, but also cyclic GMP is involved. The signal transduction process has many resonances with human vision. Nature apparently has used opsin-like proteins in many instances. The cephalopods use the same visual thing that we do, and they developed it independently of mammals and vertebrates. So the way it appears to work, and unfortunately this is sort of ended. No one that I know is pursuing this, but when light strikes the membrane, it activates these photoreceptors, and the photoreceptors are coupled to the enzyme that makes cyclic GMP, and cyclic GMP goes up, and that then directs the formation of an actin filament network, and that's experimentally verified. And I believe what happens is that vesicles are transported on these then directionally aligned microfilaments and are inserted into the membrane on the other side. And I believe those vesicles contain calcium channels, and that's how calcium comes in. But when it gets to that level of detail, that's pretty hypothetical. I cannot cite direct evidence for that. And I didn't have the resources in my laboratory to pursue that. And I wish somebody would, but I don't know. Because I think it's an interesting story. We were able to show, using fluorescent techniques, that in response to the light, we can actually see a calcium gradient with high calcium at the end that's going to grow.
But was there not a story of like a one-dimensional capillary tube or something like this, and you put the eggs in, and they were sort of polarizing each other? Am I imagining that? I thought there was a story on this.
No, they weren't polarizing each other. This was Lionel's early work to try to measure them. What he was doing was he put the eggs in a capillary tube and then shine light to polarize them, and it was like putting batteries in series.
Okay, so they were acting independent. They were still acting independently.
They were still acting independently. They were not polarizing each other.
I see.
Ken, I believe you also did a lot of work on the proton pump and took it forward. Can you talk a bit about what is the proton pump and the significance of it?
Well, that was most of what I did on proton pumps was really done in collaboration with Danny Adams. And so my interest in everything there, it really was reflecting what she was doing and doing with Mike at the time. By that time, what I had available to me, though, were methods of actually measuring proton fluxes at the level of the single cell. using a moving electrode that was ion specific. And I think that tool was valuable in being able to directly show that there were changes in proton movements during development, early development in amphibians. And furthermore, some of the drugs that interfered with left-right asymmetry also directly interfered with these proton movements, these asymmetrical proton movements across the left-right axis. So those were really fun experiments to do. But in some ways, I consider myself to have been sort of the junior partner in that. I was providing a technique to follow up on some of these ideas that Mike and Danny were working on.
But it was pretty remarkable because, you know, we were studying the way that embryos determine their left from their right, and it was very controversial at what point do embryos really know left from right. And, you know, I was arguing that it's very early, and then Ken's data directly showed us that if you watch the proton pumping, they consistently know which side is the right side by the
they do indeed. Yeah.
It's very important.
And that would be pretty early on in development, like the embryo itself would know what's the.
This was at the 2-cell stage. Okay.
Wow. So once you enter Lionel Jaffe's lab, you start doing some incredible work. What was the state of the field, perhaps like a decade later from then? What was some of the most significant research being done in bioelectricity then? Maybe this would be the 1970s, perhaps.
Yeah, that was actually a period of tremendous growth. I was Lionel's first graduate student, and even though he started out his career as a professor at Brandeis, and then he went to Penn, and then he came to Purdue, but he had never had a graduate student who completed a PhD with him. So he was sufficiently pleased with the way I turned out that he began recruiting in the physics department from their graduate students. And I don't think the physicists liked that very much, but I think he got 3 more graduate students from that. And the first one being after me was Rich Nuccitelli. And the laboratory then began to increase significantly in numbers. And so I think at one point, Lionel may have had 10 or 15 graduate students and postdocs in there. Muming Pu joined the lab as a postdoc, and he and I collaborated. And after I finished my PhD, I hung around there for another year or so, just because everything was moving so fast and there was a lot going on. And Lionel, in the meanwhile, began to look at higher level things, regeneration of amphibian limbs. And Richard Borgens came into the lab, and he was a joint graduate student with Joe Vanable, who was interested in these matters. So that stuff's changed dramatically in those years. Pu and I showed that if you expose cells to an electric field, just pass current through the medium that they're growing in, you could cause membrane bound receptors of a certain color to migrate directionally in the electrical field that we applied to them. And then I later showed, and first, and Pu then independently showed that that these same electrical fields, tiny electrical fields, could direct the movement, the direction of nerve growth in culture. And that got the attention, I think, of a lot of other people about you could actually guide neurons with small electrical fields. So there was really a huge increase in interest and number of people working on these matters in the 70s. Excuse me.
But what were some of the pushbacks then, or were there any pushbacks from other people about the field?
Oh, sure. There was resistance to the idea that electrical fields, endogenous, naturally occurring electrical fields, were playing any role in polarization. I think there was a strong bias toward thinking that all communication between cells was probably chemical, and diffusion, and depending on diffusion, and real resistance to the idea that these electrical currents that we measured and electrical fields that we could measure inside the embryos were playing a role in polarizing them. And I would say today that that's still not a settled issue at all.
So when you did work on showing that neurons respond to these electrical fields, were people's reaction that this is limited to neuron cells and it doesn't work for the other cells in the body, or even for neurons there was resistance?
Well, we showed neurons directly on neurons was in culture, okay? So, and that work was well, people believed it. The data was very sound and it was very visual, and so that was straightforward. But the resistance was that we did not show that in the intact embryo, that endogenous fields that were already there, were actually affecting development and movement, migration of neurons and migration of cells. And I must say that's one of the failings of what we did. And toward the end of my career, before I retired, I was trying to work out ways to do that directly, to do it in the intact embryo. And some of the, I was envious of what was going on in Mike's lab and what he was able to do, and I was trying to meet that. So I thought, okay, first of all, to do this, I need a genetically tractable system, and a small transparent, an animal that makes a small transparent embryo and everything. And so that led me to zebrafish. And I had hoped that we would be able to actually visualize growing neurons in zebrafish embryos and actually measure and manipulate the local electrical field inside the embryo, and see the response of the neurons as they were developing. So as a kind of, just to get going on that, we set about repeating the experiments that we had done with Xenopus in cultured neurons, taking out, and those cultured neurons were gotten from the developing nerve cord of the embryo. So we did the same thing with zebrafish. We took out the the developing nerve cord and disintegrated the cells and put them in culture and grew them and applied electrical fields to them. And they didn't respond at all. And that was a real setback in my plan, because I had just, I'm sorry, hang on just a second. I traveled to New Mexico and I got a cold, and so I'm still recovering from that. So we couldn't, they just didn't respond. And I must say that my fellow scientists, people, some of whom I trained in my own laboratory, did not like that result and its implications. So I never did. And then, in fact, I was never able to do the experiments that I envisioned, partly because we couldn't even show at the culture level that the cells were responding to electrical fields. Now, we did do some work using zebrafish on keratocytes, and they are exquisitely sensitive to electrical fields. And there are some people continuing to work on those aspects of it, whether endogenous electrical currents are involved in guiding wound healing in these embryos, in these organisms.
And what was it like in Lionel's lab? I mean, it was like an incredibly productive period. So much good stuff coming out. Just some real characters, as far as I understand. What was that like, for you and in general?
Oh, it was a blast. There was a lot going on, and as you say, there was a colorful cast of characters, and some amazing science that just hanging around. Totally separate from the electrical field stuff going on right there. I wasn't part of it, but I was witness to it. Was the measurement, the first measurements of calcium inside cells of using optical techniques, using the photoprotein aquarin, the bioluminescent protein aquarin, this was before its accessory protein, GFP, had even been discovered. This is just purified aquarin from a jellyfish. And aquarin, when an aquarin molecule binds to calcium ions, it then emits a photon, and it's dead at that point. And so Lionel had the vision to inject aquarin into a fish egg, Medaka, a clear fish egg, and then using a photomultiplier tube, measure light output when the egg was fertilized. And I very well remember when they were doing, they have these extremely sensitive photomultiplier tube, and the egg is sitting right on the cathode of the photocathode of the photomultiplier tube. And they added sperm in a completely dark room. And the signal that was being recorded electrically, it simply, it just blew it off. They had to change the gain by 3 orders of magnitude in order to keep the signal on the screen. And Lionel then went to the astronomers, and they had imaging devices that worked at the photon counting level. And so he collaborated with those people, a group at Princeton, and then was able to actually visualize the calcium explosion that occurs when the egg gets fertilized. You could see it start at the point of sperm entry and then spread around the egg. And that turns out to be a universal truth. All eggs, I know of no exception to this, plant, animal, algae, it doesn't matter. At fertilization, there's an explosion of calcium that is both necessary and sufficient to start the developmental process. And that was being done right. I was right next to it. I didn't do that work, but I was witness to it. And that was truly an exciting era in science.
What do you think it was? What was Lionel doing that enabled this? Like just all together, this amazing fertility, the creativity, all these great people. Was there anything in particular you would attribute that to?
I would say the defining characteristic was Lionel's absolute pure curiosity. And, I remember once somebody was doing some work and the results were absolutely not what was expected. And they sort of shot down a major idea of work. And I remember Lionel's reaction to that was so matter of fact and accepting. He wanted to make sure it was right, but okay, we scrap that and we start all over again. And this actually happened to me. I'd be working away in the lab and everything, and late at night, and something, I get some exciting results or something like that. I could call Lionel up at any hour of the day or night and get a conversation, and he would just be so eager to talk about it and work through it with me and everything. He just had a kind of almost a purity of perfect scientific interest in everything. Mm. Now, on the other side of the coin, Lionel had, I think there's terminology for this now, but I don't know if it was then. Lionel did not have a good grasp of the impact of what he said on other people. He couldn't calibrate, and he could be brutal. And I think mostly not intending to be harmful. It's just he just didn't recognize what effect he was having on people. So he made a lot of enemies, and was not widely beloved in the field. And I think that was a severe enough problem that it interfered with actually the perception of his work and the acceptance of his work. I think it had an impact on that. It's interesting, his daughter, Lorinda, with whom I collaborated some, and she did, and I was responsible for getting her first job at the University of Connecticut Medical School, where I was at the time, and she's still there. She was not too long ago elected to the National Academy, and Lionel never was at all. And I think in part this was personal, and his personality interfered with things. And
Did he also?
sorry, go ahead.
No, go ahead.
No, I I was just going to mention that the last 5 years of Lionel's career in science were pretty, he had no grant support. He didn't even have a laboratory to work in, and it did not end well.
Did he also try to instill some sort of camaraderie, or how did he have certain ways of motivating people, or was he mostly leading by example through his sheer curiosity?
Well, I think mostly he was just, first of all, he was a font of ideas. I mean, he had an understanding of physical principles that was just extraordinary for somebody who was trained as a botanist. I mean, Lionel was brilliant, and he had absolutely no difficulty with the mathematics and physics of what he was doing, but his insights, physical insights, generated so many ideas and he put those out there, and so it was exciting. And he was accessible. You could talk to him, and he would tell you, and you could work through ideas, and he had ideas about actually how to do experiments, which were sometimes not very good. Something that both Rich Nuccitelli and I brought to Lionel's laboratory was maybe better laboratory skills than Lionel himself had. He was held back by being overly cautious in the laboratory, I think. Things had to be absolutely perfect. And I'm thinking about the development of the so-called vibrating probe, which you could first measure currents at the single cell level and everything. Lionel had this idea for a long time and had played around with it and everything. And I don't think it would have ever gotten built and turned into a functional instrument without Rich Nuccitelli doing it, because Rich had a real knack in the laboratory of how to put things together and make them work, more so than Lionel did.
Yeah, we spoke to Rich as well, and he also had very fond memories of working in Lionel's lab. I know you and Mike have also collaborated on quite a few projects. What have some of those been, and what's that been like?
Well, yeah. I first knew Mike when I got this large packet in the mail, and it was a bibliography of everything that had ever been written in about bioelectricity, and he asked me to look at it, and I'd never heard of it. I think Mike was at that. I don't know if you were a graduate student, Mike, or a postdoc.
I was an undergrad. This was
well. It was an undergrad. Yeah. Okay. Well, and I looked at it and I was astonished by it, that it was so absolutely complete, and I wondered what had motivated this guy to put this all together. But at any rate, then we later met at a scientific meeting, and by that time you were, I think, a postdoc, and you started telling me about your ideas on left-right asymmetry and in the chick embryo and everything. And I thought, you know, there's something I can contribute to. So we started a collaboration, and I had a Swedish postdoc who came to my lab fully funded from the Swedish government. And we were looking for a project for him. So I suggested that he do this. So the idea, what we wanted to do was to look across the midline of the developing chick embryo, the left-right midline, and look for differences in the membrane potentials of the epithelial cells on one side versus the other. And my first idea was to do this with electrophysiology, microelectrodes. So stick microelectrodes in one side and then the other side. And that turned out to be impractical. There was too much noise in our measurements, and it wasn't working. And so for once I had a good idea, I thought, okay, this is a planar situation. There are a bunch of cells here. They're all the same. The only difference in these epithelial cells is they're on one side or the other of the neural tube. And so I thought this would be a good place to use imaging. And there are these so-called slow membrane potential dyes. They're really not measuring membrane potential directly. They're not responding to membrane potential, but rather they accumulate in the cells according to the membrane potential of the cell. They just distribute across the cell according to the membrane potential of the cell. And we could load these cells, this epithelial sheet, expose them to the dye, and then look how much of the dye was taken up by the cells as just with one confocal image. And we did that, and it jumped out that it was very clear that the membrane potential of the cells on one side was different than the membrane potential of the cells on the other side. And furthermore, some of the reagents that Mike had found interfered with left-right asymmetry, also destroyed that electrical potential asymmetry. And you know, all of the work that Mike had done to that point added to what we could add with the imaging and everything, made for you know, I think quite a remarkable paper, the paper that we published in Cell. That was an awful lot of fun to do those experiments. Unlike most of the time, I actually had an idea that worked out, and wow, that worked. So that was fun. And I think that also was a useful thing for the whole field, because, you know, Mike and Nanny Adams went ahead and made use of that technique in a lot of other circumstances.
You know, just following up on one thing, you point out that this was an epithelial kind of situation, and you had this amazing paper long before that about the epithelium as a generator of developmental information.
Yeah.
And if you could just talk for a few seconds about that, because I think it's a really profound notion, you know, that information for these things gets generated on a 2D surface, and then is able to instruct, you know, deep three-dimensional structures underneath that. I found that very, you know, kind of very inspiring. Anything you want to say about the kind of the epithelium and the role of it in general?
Well, I guess what I would say is that the epithelia of animals are just so powerfully electrogenic. I mean, that's what they do, is they are to the embryo what the plasma membrane is to a single cell.
Mm.
They separate outside from inside. And they do this by transporting ions. They pump sodium in and develop not a membrane potential, but a transepithelial potential. And if you make a hole, any kind of a hole in that epithelium, current comes rushing out of that hole. I mean, it's really, it's so easy to measure it. I used to do a demonstration in my undergraduate teaching. I had a very sensitive galvanometer, current measuring device, and I would take my two fingers, and dip them into beakers that were then electrically connected to the galvanometer. And I do this for undergraduate students, and just by two fingers, and you put your fingers in it, and not much happened. The meter flopped around some, but then I would take a razor blade and just make the tiniest nick in one of my fingertips and put it in, and whammo, the current, it was just really obvious. The current was pouring out of the cut surface, the cut wound. And that's the external current that's looking on the outside. But those same currents flow in closed loops. So there are also currents flowing inside the embryo. And furthermore, it turns out that you don't just have, it doesn't just happen when you make a nick in it. It happens during embryonic development, that there are places of low electrical resistance through the epithelium. And the embryo then does drive huge currents. I'm talking about the frog embryo, but it's true of other embryos as well. And I still believe, and we have reasonable evidence to support this, that those currents are involved in organizing the development of polarity and inside the embryo as it develops. Because if we interfered with those currents, or made ectopic sources of currents and everything, then you got profound effects on the subsequent development of the embryo. This is work I did with Kevin Hoteri, and a couple of really nice papers, I think, came out of that work on both chick and frog.
Just some closing. Go ahead,
Sorry.
Ken.
Go ahead.
Yeah, I was going to jump to another question and ask you just some closing questions. What are some of the advances you'd like to see the field of bioelectricity make?
Well, I would like to, the major thing I would say is that I would like to have people have it demonstrated unequivocally that these currents that I've just been talking about are in fact guiding cells and affecting behavior of the cells in the intact embryo. Either through imaging or whatever techniques, but that has not yet been done. I mean, we showed on a macroscopic level, if you will, of the whole embryo, that if we interfered with the currents, we interfered with development. But we didn't show what the specific targets were of these currents and really in fine detail where they were working. And so to do that, to show directly, okay, at this stage of development, these currents are guiding these cells to do thus and so. If that would be my goal, if I were working, that would be what I would want to see that developed, and that has not been done.
A question that I wanted to end with is there anybody in this field that you think needs to be more known about? Who, if anybody, hasn't gotten credit? I mean, we know the major players that we talk about, but like, is there anybody that you feel hasn't been heard about?
Well, I would point to one person, and he's not unknown, but he was a former student of mine and graduate student and a postdoc as well. And his name is Mark Meserly.
Mm.
And he's in South Dakota State University. And he is really doing I mean, he is a biophysicist par excellence. And he is really doing work on understanding the mechanism of electrical fields and their interactions. And particularly what he's focusing on is the role of electrically induced water movement in cells, and doing just fabulous work, I think, on that. And I would like to see Mark's work better known and better recognized.
Cool.
All right, Ken. Thank you so much. Is there anything you'd like to add as we close out?
No, I think I've said everything I have to say. It's been a pleasure to meet you, and really fun to talk to you again, Mike.
Yeah, great to see you, Ken. Thank you so much. Yeah, amazing.
Okay. Yeah.
Thank you so much for your time.
Super.
All right.
Be well.
Bye.
Bye, everybody.
Adios.